recording. And here we go. So we talk about species and speciation. I'll take you back to 1928 with a young German ornithologist named Ernst Mayr, shown here on the right with his assistant Sario on the left, a Malaysian gentleman, Sario. No record if he was related to Mario or Wario or Luigi or anybody like that. Anyway, he's gone off to New Guinea to collect and classify a bird family known as the birds of paradise, the Paradiseidae. And he's studying some other tropical bird families in the region. The motivation behind this, by the way, other than the thrill of scientific discovery, is that birds of paradise had actually were actually in demand at this time because their feathers were exported from New Guinea and used in the manufacture of ornate ladies' hats. This is still the time when, ladies wear, I guess hats in general have kind of fallen out of fashion, but big hats with ornate feathers and things like that were still popular at the time. And there was something of a global industry in New Guinea tribesmen, New Guinea folk would shoot birds of paradise and export the feathers to Europe into the hat making industry. And there was some interest into, what sort of species these were that were producing these feathers, because there were a lot of different forms, some of them with extremely long feathers. I think some of the longest feathers in the world are made by New Guinea birds of paradise. So Mayr went there and ended up shooting over 3,000 of them, to which I will just say it was a different time. These days, we try to study biological diversity without shooting quite so much of it. But you could do that back then. Here you can see the ornate feathers. And Mayr was baffled by the diversity that he found. There's a huge number of different forms of birds of paradise, and it wasn't clear how to divide them. This is technically not a bird of paradise. This is in a different family, but it's another tropical bird that comes in a lot of different varieties. You can see they've got different feather crests on the head, and they varied a lot over their geographic range, but there didn't seem to be an obvious way of neatly dividing them up. Ever since the days of Linnaeus, there's been a need for classifying living things. I assume you've heard of Linnaeus. He was the Swedish botanist who came up with the system of binomial nomenclature and the taxonomic hierarchy that we still use. His original motivation was that he was a doctor. Doctors had to prescribe medicinal plants. That was the source of most of the drugs they used. And if you misidentify a plant and you give it to your patient, you could end up killing the patient or at least not doing any good. And if you have hundreds of medicinal species and that number is growing every year, you have to have some sort of indexing system to keep track of them. That was the original motivation for coming up with the classification system that we use. And Mayr was having a hard time applying it because there's so much variation, but it's not immediately obvious how to divide it up. Forms tended to blend into each other, or two forms that were very different in one way might be identical in some other way. And what Mayr developed as a result of his attempt to study tropical birds of paradise is the biological species concept. And this is what you have probably been taught as the official definition of species. You define a species as a set of potentially interbreeding populations Two species are separate if their members cannot successfully interbreed. And by successfully interbreed, I mean can reproduce with each other and produce healthy and fertile offspring. So, to take the example that you've probably seen in your textbooks, horses and donkeys are considered separate species because they can interbreed, but the offspring are sterile. The offspring, of course, are mules. But if you have populations that can interbreed, or at least potentially can, you place them in the same species. That means, by the way, that the biological species concept doesn't line up with how species necessarily look. If you've taken ornithology, you might know that things like the Carolina chickadee, which I think is the one we get here, and the black-capped chickadee to the north have different calls and they don't interbreed, but they look virtually identical. You can't really tell them apart by looking. And the same is true with things like the western and the eastern meadowlark. On the other hand, there are some species that can be extremely variable in appearance, and yet all interbreed. Humanity's one. We come in a wide range of, you know, colors and shapes and hairstyles and things like that, and yet we're all potentially perfectly capable of interbreeding with each other, and in fact have been doing it for many thousands of years. Meyer went a little further than this and proposed a model for how new species form, and it's allopatric speciation. Patria is the Latin word for your fatherland. So if you love your patria, you are patriotic. That's where the word comes from. Allo happens to be the Greek root meaning different. So allopatric means different homeland. As we'll see, you can also have speciation within one homeland, and that's called sympatric speciation. And then there's a third form of speciation called St. Patrick speciation that's only been documented in Ireland. I hope nobody wrote that down. That was a joke. In allopatric speciation, you start with a population of one species, a group of individuals living in the same area, connected by common descent and potentially interbreeding amongst themselves. And that population gets split by a physical or geographic barrier that members can't easily get across. Exactly what this barrier is could be, I mean, it very much depends on the environment. If you, if you've got species that are living in a river, if the river changes its course and leaves a meander behind or something like that, that could divide the river species into separate populations. There was a person at Hendrix for a while who worked on little relatives of rabbits called pikas living on in mountain ranges in the American West, and populations in different mountain ranges used to be able to interbreed or used to be able to mingle, but because of climate change, the valleys between the mountain ranges were now too hot. So you could have a change in the climate cutting off means for populations to mingle. It could be a change in a river course. It could be it could be a mountain range coming up. It could have to do with plate tectonics, which we'll talk about later. In some cases, it can even be the building of a major highway. They found genetic differences between populations of garter snakes on either side of Interstate 10, because that's a barrier that snakes cannot easily cross. Anyway, If this happens by organisms migrating to a new habitat or being carried to it, we call it dispersal. If the organisms don't move themselves and the barrier just splits them, we call that vicariance. Show you some case studies of this in just a bit. Dispersal and vicariance. Dispersal, the organisms do the moving. In the case of vicariance, the organisms don't move and the barrier appears around them or appears within them and divides them. Those separate populations are now going to tend to become different. If they end up in different environments, they will be subject to different selective pressures. Natural selection will tend to make each different from the others. Alleles in one population can't be exchanged with the other. So if we get any mutation producing new alleles in one population, those new alleles won't make it into the other population's gene pool. We say that gene flow is blocked. Genetic drift, this is not on the slide, but genetic drift will also tend to make those separate populations different from each other, just because allele frequencies in one will drift away from allele frequencies in the other because they're no longer exchanging members. And then finally, one population may end up with an unusually high or low frequency of certain alleles purely by dumb luck. And we call that the founder effect. This is particularly true in cases where a new population is founded only by a small number of individuals. And I'll show you some great case studies of that in just a bit. Anyway, all of those, selection, drift, gene flow blockage, and the founder effect, will tend to make those two populations different from each other, and the difference will tend to increase with time. Given enough time, the idea is that the separate populations will diverge so much that they'll become separate species, and even if the barrier disappears and the species come back into contact, they won't interbreed. If they come back into contact and they do interbreed, the separate populations will just kind of fuse into a single one again. But if they come back into contact and don't interbreed, then we have separate species. There's some nifty examples of the founder effect in human genetics, and these have not yielded separate species. We're all still, all of humanity is still one species, and humans from all parts of the globe are perfectly capable of interbreeding with each other and have done so throughout history and before that. But we still have cases where a human population that settles in a place and is somewhat isolated from surrounding populations can show the founder effect. These are the hands of a person with a disease called porphyria. It's a disorder of heme metabolism. You might remember that your hemoglobins contain this ring-shaped functional group called heme that bonds with the iron atoms that hemoglobin needs. And if you can't metabolize heme properly, you have porphyria, which can cause skin disorders, can also cause mental disturbances. King George III, who you probably know as the guy who sings, "Oceans rise, empires fall, we have seen each other through it all." Anyway, George III of England had periodic bouts of insanity. I've actually seen the palace and the gardens in the south of London, where they would put him when he was in one of his fits and would ride around having screaming matches with the trees. Anyway, it is suspected that he had porphyria. Oh, I almost forgot. Erasmus Darwin was asked to treat the king for his insanity, but turned down the job. So little connection there. Anyway, there's another form of porphyria that happens to be unusually common among South Africans of Dutch descent, the so-called Cape Dutch. And the reason is that only a small Dutch population settled there. One of the members who's been traced, we have very good genealogy for the Cape Dutch, and we actually know who she was. She was a female orphan named Adriansa, who came over in 1688, and she just so happened to be a carrier of porphyria. Porphyria is relatively rare in humanity on the whole, but just by stupid luck, she happened to be a carrier of it. She had kids. The kids had more kids. Given enough time, eventually you will have distant descendants of Ariantia marrying each other, and then you get people who end up homozygous for the porphyria allele. I don't have a slide for it, but I grew up about maybe 30 miles away from a little town in South Louisiana called Kinder. And in the 1980s, there were some cases reported of babies that were born with this very unfortunate congenital disease, where after, I think, about a year, they would just quit thriving. They'd start losing motor control. They'd never develop intellectually. They were usually dead by about the age of 2. And this was just one of those things, back, 100 years ago, babies died because, medicine couldn't really treat them. So it had probably been going on for at least a century. And it wasn't until the 1980s that someone realized that what it was Tay-Sachs syndrome. Tay-Sachs is a disorder of lipid metabolism that causes toxic byproducts to build up in the brain and causes irreversible brain damage. And you usually die between, I think, about 2, maybe a little longer. I think maybe between 2 and 4 is usually when it claims a life. Tay-Sachs is very rare, except it happens to be relatively common among Jews of Northern European descent, so-called Ashkenazic Jews. But one of the French-speaking settlers who had been expelled from French-speaking Canada in 1768 and had found their way down to South Louisiana and become the Cajuns, one of those settlers just happened to be a carrier of Tay-Sachs syndrome. Maybe he had some distant Jewish ancestry, I'm not sure. But he had kids and they had kids, and eventually distant cousins start marrying. That's inevitable in a small population like that, even if you're not trying to inbreed. And eventually babies started being born with 2 copies of the Tay-Sachs allele. And this had been going on for some time, but it wasn't until the 1980s that it was identified for what it was. And that's another founder effect. Like the Cape Dutch, the Cajuns of southwestern Louisiana were fairly isolated from the mainstream until about the 1930s. There weren't many roads in. Everybody spoke French. There just wasn't that much cultural contact with the rest of the country. One more. In 19th century America, congenital deafness affected about 1 in 5,700 people. The island that you see at the lower left is Martha's Vineyard. Next to it at the lower right is the island of Nantucket, which is famous for its role in a great poem that goes, "There once was a man from Nantucket who kept all his cash in a bucket. His daughter, named Nan, ran off with some man, and as for that bucket, Nantucket." That's it. Anyway, Martha's Vineyard over there Today, Martha's Vineyard has some of the most expensive real estate in the country. It's a place where, Silicon Valley big time CEOs might buy vacation homes on Martha's Vineyard. Um, it's a beautiful place, great place to vacation, um, very, very expensive real estate. Up until fairly recently, though, it was pretty isolated culturally from the mainland. There were people living on it. They farmed. They did a lot of fishing. They tended to keep to themselves. And on the islands of Martha's Vineyard in the 19th century, the incidence of congenital deafness was as high as 1 in 155. And in one town, it was as high as 1 in 25. 4% of the population was born deaf. And this is also the founder effect. A few of the colonial settlers, Europeans had been settling there as far back as the 1600s. And a few of those settlers were either deaf or carriers of an allele that caused congenital deafness. There's actually more than one way in which you can be born deaf. And evidently, genes that have that effect just happen to be present in the founding population of Martha's Vineyard. And so this deaf community grew up on Martha's Vineyard. There were actually a couple of other New England towns that had unusually high deaf populations. There was one in Vermont. I think it might have been might have been Stowe, Vermont. I'd have to look that up. But the reason, again, is founder effect. Small groups of settlers founded those towns. They tended to be cut off from the wider world. And you end up with this deaf population that's relatively large. And this is historically important because the Martha's Vineyard deaf community was not cut off from the rest of the island community. In a lot of places at this time, the deaf were, institutionalized or just locked up at home or something like that. But with this many deaf people on Martha's Vineyard, they built a community and they developed a way of communicating with signs. And even hearing people on Martha's Vineyard learned to communicate in sign, mostly because, if you own a store and a bunch of your customers are deaf and they've got money that they want to spend, well, you need to learn to communicate with them. And so Martha's Vineyard Sign Language is one of the roots of American Sign Language. When a bunch of students from Martha's Vineyard and these Vermont towns came together in a school, and they were taught by an educator who'd been trained in France and learned Old French Sign Language. What grew out of that was a combination of French Sign Language with Martha's Vineyard Sign Language. And that's ultimately where American Sign Language comes from. It's not just English spelled out with your fingers. It's a completely different language all its own with very different roots. All right, so much for humans. Here's a non-human example. This is the Argentine ant, lives in colonies in South America, and ants from different colonies attack each other. And they attack each other because ants from different colonies have different waxes, different hydrocarbons on their exoskeletons. So if two ants meet and they taste wrong, they'll fight, or they taste different, they'll fight each other. What's happened is that the Argentine ant has been introduced to California, Japan, and Europe. And in part because of the founder effect, the ants that have been introduced to these places are so genetically similar that they taste the same and they don't attack each other anymore. So instead of having a bunch of little separate colonies that fight, colonies of Argentine ants in places like California all descend from one or a very small number of founders that got introduced to California. They've all got the same hydrocarbon waxes on their exoskeletons. They all taste the same to each other. And the result has been the development of super colonies, these interconnected multi-queen colonies that can freely exchange members. You can see where they've been introduced. You can see California is basically one big Argentine ant super colony. So is the Gulf South and Atlantic coast of the US. The largest is that one that extends along the coast of the Mediterranean, from Portugal all the way to Greece, extending 3,600 miles. Okay, so that's what the founder effect can do. Couple that with genetic drift, selection, and blockage of gene flow, and separated populations will tend to become different from each other. You can separate those populations by either dispersal or vicariance, and the Galapagos life is probably a great example of dispersal. The Galapagos Islands have never been connected to the South American mainland, right? They're 600 miles away from land, and they're pretty young islands. They're still volcanically active. Volcanic lava flows and things still do happen there. There's no geological evidence that there was ever any kind of land bridge between the Galapagos and the mainland. So what must have happened is that Galapagos life must have come from South American life that dispersed across that 600 miles of water. We may never know the precise details. Finches don't normally cross 600 miles of open water, right? They're not long distance birds like that. We may never know exactly how they got to the Galapagos, but it's possible that some South American finches got blown there in a storm. It's possible that some small tortoises drifted from South America to the Galapagos Islands, maybe on a log, maybe on a tree that blew over and drifted out to sea. We've actually, maybe I'll get to this later. There's some cases in the modern world where we've actually been able to observe that happening. However it happened, South American finches must have dispersed to the Galapagos, crossing a barrier that they don't normally get across. And then finches must have dispersed again from one island to the next. And so you have a whole bunch of little separated populations, each of which has given rise to a separate species. So that would be speciation, allopatric speciation by dispersal. On the other hand, my favorite fish in the whole wide world are desert pupfish. If you go down to Death Valley, California, you really should. It's an amazing place. And get yourself a map at the main park office at Furnace Creek and drive a bit northeast past the Beatty Cutoff Road, and then turn off and go down a dirt road, and you can be at a place called Salt Creek. Now, this is some of the harshest desert in the world. The record high temperature measured at Death Valley was, I want to say, like 100. I want to say it was 138 Fahrenheit. I may be underestimating that. But there are a few places in the Death Valley country where there are springs that do feed permanent pools of water. And at Salt Creek, which is fed by a spring coming up out of the ground, putting out salt water, you have this little native fish that you see up at the top. That's the Salt Creek pupfish. Drive around to the south end of the park at a place called Saratoga Springs, and you can't really get close to the water there. But if you could, you'd see a different pupfish species in the spring-fed pools at Saratoga Springs, called the Amargosa pupfish, which is the one that you see down there. And in fact, there are little pupfish populations all over 300 miles or more of California and Nevada desert. What's happened here is not dispersal. What happened here is that 10,000 years ago, the climate was much cooler and much wetter. And what's now very harsh desert was filled with a system of rivers and lakes. And presumably, a pupfish species got in there and managed to spread all through that system of rivers and lakes. What's happened is that the climate has become hotter and the climate has become drier. The rivers that used to flow have almost entirely dried up. There's a few rivers in the Death Valley country. The biggest river, the Amargosa River, does not actually have any water in it for about 11 months out of the year. So I don't know if it really qualifies as a river. So anyway, that's dried almost all the time. The ancient lakes in the desert country have now completely dried up, except occasionally they'll refill during El Nino years. But most of the time there just isn't anything there. It's just barren salt flat. And the only places where the lake life has survived is these little spring-fed pockets. So this is vicariance, because what happens is the fish didn't go anywhere. Little inch-long fish can't really cross 100 miles of some of the harshest desert in the country. What happened is the fish stayed pretty much what they are, and changes in climate caused the connection between these isolated populations to disappear. There's now no longer any way that a pupfish at Salt Creek can swim to the pupfish at Saratoga Springs. The connections all got cut. So that's vicariance. The fish have stayed in place, and the connections between these populations have been severed. Barriers have appeared. In this case, the barriers are, 100-mile stretch of very harsh desert separating these little refugia, these little pockets of water and wetlands habitat. So yeah, if you ever get out there, you really have to go check out the pupfish. It's our own little Galapagos Islands, and it's practically in your backyard. Well, if your backyard is 1,500 miles long. Have we ever seen this happening in real time? Well, in 1964, a lab in Long Beach, California, started raising clamworms. Real quick, that's a clamworm. These are segmented worms living in the ocean, popular for fish bait out there, I'm told. You can see they've got a segmented body like an earthworm, but every segment has a pair of little paddles called parapodia, and fairly easy to raise in an aquarium. So in 1964, the Long Beach lab started raising a population of captive clamworms. 1986, 4 pairs of these worms were brought from Long Beach to Woods Hole, Massachusetts, and raised at the Woods Hole Oceanographic Institute, WHOI. 1992, somebody got the bright idea to take some Woods Hole worms and try to crossbreed them with wild worms that had been collected at Long Beach, and they failed. What happened was, first of all, this is one species where there's male parental care. The males build these tube-like nests. The eggs are laid in the nests, and the males will sit on top of the eggs, protect them against predators. They've got pretty big jaws. And males will also fan the eggs with their little parapodia, those little flaps that they've got. to keep a steady current of water flowing over the eggs so the developing embryos get oxygen. Males wouldn't take care of hybrid eggs, so hybrid eggs died. Well, you can artificially rear Nereus eggs by putting an air stone nearby, so you get, a stream of air bubbles passing close to those eggs. That will give them enough oxygen and enough current flow that they can develop without a male. But even when this was done, hybrid eggs failed to develop. There's some kind of genetic incompatibility between the Long Beach wild population and the Woods Hole captive population. So by the biological species concept, what we now have is two worm species. They don't look different from each other, so they're what we call cryptic species. Like the examples I gave you earlier, the black-capped and Carolina chickadees, or western and eastern meadow larks in among North American birds. Those are also examples of cryptic species, separate biological species because they can't interbreed successfully, but they don't look that different from each other. I don't know the details, I should probably look this up sometime, of exactly why the Woods Hole and the Long Beach worms are somehow genetically incompatible. But when we talk about the biological species concept, we can talk about different isolation mechanisms. The biological species concept is based on reproductive isolation. And isolation mechanisms can actually happen at multiple different points in the life cycle. Sorry, checking the chat right there. And we can talk about prezygotic and postzygotic isolation mechanisms. Prezygotic is due to factors that operate before the nuclei of the sperm and the egg fuse. So species can be isolated behaviorally, in which they don't interbreed because they've got different mate recognition behaviors. If one population of birds goes tweet, 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 and the other goes chirp, 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 then males of one may simply never recognize females of the other, or vice versa. Females of each species may simply just not recognize males of the other species as potential mates. If you have two anolis lizard species and one of them has a bright red dewlap and the other one has an orange striped dewlap or something like that, females of one species may simply not register that they can mate with males of the other. So we'd count that as a reproductive isolation mechanism. In temporal isolation, individuals don't interbreed because they mature at different times. So you might have two species of, I don't know, plants that maybe you could try interbreeding them, but one of them flowers in April and one of them flowers in June. And so, pollen from one will almost never find its way to eggs of the other. You can have ecological isolation where individuals don't breed because they're occupying different niches. You can have gamete level isolation where individuals don't interbreed, sometimes because the gametes are just biochemically incompatible. The receptor proteins on the sperm don't bond with receptor proteins on the egg, maybe. You get this a fair amount in plants where pollen of one species will simply not germinate, won't grow a pollen tube if it's on the stigma of a different species. So, that would be another form of gametic isolation. There could be some sort of chemical incompatibility between gametes and parental tissues. I guess that pollen failing to germinate would be one good example. In some species of animal, you can get mechanical isolation. Um, Insects have very distinct genitalia, or many groups of them do, and it's not always possible to identify species unless you get a very good look at the male genitalia. This is so important that an Australian company in the 1990s invented a device that they sold to entomologists that used very low pressure pumps pumping fluid through an extremely fine needle to inflate the male genitalia of insects so that these could be seen and described. They called it the phalloblaster, which I think is one of my favorite names ever, the phalloblaster. That's important if you have to identify insect species, because there's many cases where insects can only be told apart by genitalia shapes. If you do systematic entomology in a big way, you're going to look at a lot of insect peen. And this is also true for crustaceans and other arthropods. And the reason it's important is that male and female sex organs fit like a key into a lock. Different species may not be able to interbreed simply because the male's key does not fit the female's lock. There's at least one case I'm aware of where an attempt at hybridization was documented ending when the male's genital appendage snapped off, which pretty much took both the male and the female right out of the gene pool. Kind of hard to mate if you've got the tallywhacker of a male of a different species permanently blocking your orifice. Ew, yuck, sorry. Probably should not have phrased it that way. But you can have mechanical isolation in cases like this, and that's also prezygotic. It operates before the sperm and the egg nuclei have joined. Postzygotic things that can happen would be the zygote the egg might be fertilized, but the zygote may be unable to develop any further. Or the zygote might divide a few times. It might eventually give rise to an embryo, but for whatever reason, the embryo just stops developing, and after a certain point, it just dies. You could have cases where the embryos do develop into individuals that are born, but the hybrids between species might be sterile, like mules, where the hybrids may survive, but they're unable to breed. And then you might have reduced hybrid fitness, cases where hybrids can be born, but they may be unhealthy, they may be weak, they may have a high mortality rate, they just may be very strongly selected against. So these are mechanisms that tend to keep biological species isolated from each other. In cases where you have two populations that hybridize, but the hybrids have low fitness, selection will tend to favor mechanisms that decrease the incidence of hybridization. If you belong to species A and you don't clearly signal that you are in species A and you want to mate with species A, then you may end up mating with species B and your offspring will either not exist or they'll be selected against. That means that there should be selection favoring individuals that strongly prefer their own species and individuals that strongly advertise what species they're a member of. If you're able to signal, hey, I'm species A, I only want to mate with species A rules, species B drools, you'll leave more offspring than if you're signaling, hey, whatever, it's all good. We call this speciation by reinforcement, speciation that's driven by selection against hybrids. It's also called the Wallace effect, after Alfred Russel Wallace. And there's been debate as to how often this happens in nature. It has worked at least in lab studies. Back in the 50s, it was shown to work in laboratory populations of fruit flies. But there seem to be some cases in the real world where it works. And this is the collared flycatcher. I believe that's a European bird on the left, and the pied flycatcher on the right. Hybrids between the two, male hybrids can breed, but female hybrids are sterile. And I should mention there's a number of cases where hybrid sterility affects one species, one sex, but not the other. That's just one of those things. Well, maybe I can talk later as to why that happens. But hybrid sterility will often affect one sex, but not the other. And in this case, female hybrids are sterile. So if you hybridize, then roughly half your offspring are not going to reproduce. Whereas if you mate within your own species, all of your offspring are going to reproduce, or at least potentially they can. Put it another way, if you hybridize, your probability of having grandkids gets cut in half. And in the areas where the two overlap, the species are more different from each other in male plumage and in female preferences than if you compare them from areas where the species don't overlap. So if you take a collared flycatcher from an area where it's the only bird and a pied flycatcher from an area where it's the only bird, they don't look that different from each other. If you take a collared and a pied flycatcher from an area where they're living in the same region, they will look more different from each other, because in the area where they overlap, it's important to signal what species you are, because if you don't clearly signal which of the species you are and which you want to mate with, your chance of having grandkids gets cut in half. You're selected against. So selection in this case has favored the species becoming more different from each other. So that seems to be an example of speciation by reinforcement, speciation by selecting against hybrids. You may have several of these barriers operating at once. Up there at the top, that's a domestic cat. That's not my cat, but it looks quite similar. That's your typical mackerel tabby. And there was an experiment done a few years ago where they bred domestic cats with servals. That's a small wildcat native to Africa. You can see one down there, Leptolurus serval. They've never been truly domesticated, but you can tame them. Somewhere I've learned the trivia fact that the Egyptian pharaohs actually kept pet servals and trained them as hunting animals. And somebody a few years back tried crossbreeding domestic cats with servals. These are not just different species, they're currently classified as different genera. And it turned out that crossbreeding them was difficult for several reasons. One is they were behaviorally isolated because servals would often refuse to mate with domestic cats. The gestation periods turn out to be different. The serval has a longer gestation period, and hybrid litters were often born prematurely or aborted, or in some cases absorbed. Humans, as far as I know, don't do this, but a number of mammals have the ability to absorb embryos back into the mother's body. Rabbits will do that, especially if you have a pregnant rabbit that's stressed. She may just reabsorb her embryos and never give birth at all. Evidently, cats can do this sort of thing too. So we have several different isolation mechanisms, prezygotic and postzygotic. And yet, it was possible to produce F1 hybrids. It usually didn't work, but it worked a few times that when you crossed a domestic cat and a serval, you got F1 hybrids. And the F1 hybrids turned out to be fertile. And the F1s were still a little bit too wild in behavior for most cat owners to be able to keep. So the F1s got back crossed with domestic cats. So you got an F2 generation that was, 3/4 domestic cat and 1/4 serval. And then after a few generations, you have F4s and F5s. And F5s are considered a breed of cat called the Savannah cat. This is one of them. And F4 and F5 hybrids, which might only have what, 1/16 or 1/32 serval genes, have the wild appearance, that spotted coat, that wild animal look that makes them desirable. But they also behave like domestic cats, at least enough that the average person can care for them. They don't do, they're not the sort of things that will completely tear your house apart just because they're bored. They're well enough behaved that they can be kept like any domestic cat. So we've got isolation mechanisms, and yet this raises the question, are servals really a separate species from domestic cats? There's isolation mechanisms that prohibit breeding between them, and yet they're not absolute cutoffs. It is still possible to interbreed them. It's still possible, at least some of the time, to get fertile F1 offspring, and those can go on to breed with domestic cats. And we have this breed called the Savannah cat. There's a breed of domestic cat called the Bengal, which was created in the same way. Domestic cats were crossed with a small species called the Asian leopard cat. And then the hybrids have been back crossed with domestic cats to produce a breed that is genetically mostly domestic cat, that is well enough behaved that you can actually keep them in the house, and yet has that beautiful, wild, uh, spotted and striped coat pattern that makes them very desirable. That's a Savannah cat again, by the way, showing off that beautiful wild type coat. And when this happens, we call it introgression. What's happened is just that one population of domestic cats has had a little trickle of serval genetic material that has introgressed into the domestic cat genome. Currently, it's only Savannah cats that have this, but given enough time, somebody's, unneutered male Savannah cat is going to get out of the house and go on to impregnate all of the female cats in the neighborhood. And given enough time, I'm sure we're going to start seeing, all sorts of domestic cats, even, stray cats that have just a little bit of serval genetic material in them. It will eventually spread into the larger cat population, much like the Siamese trait did. Siamese cats were originally, only found in the country of Thailand. When they were introduced to this country, it was a very rare and exotic breed. And then somebody's unfixed Siamese cats got out one night and shagged all of the female cats in the neighborhood. And now we have that Siamese trait all over the place. Our kitten has the Siamese patterning on her calico coat, which makes her look really striking. And so just as the Siamese trait has spread away from the Siamese cat breed and into the cat population at large, the same thing will eventually happen with those serval genes that give you that beautiful spotted coat. That's introgression, genes from one species trickling into another. And this happens even in wild species. And here's a great example. There's a polar bear at the top right and a brown bear at the lower left. We normally consider these to be different species, but in zoos, you can get them to hybridize and they'll produce fertile offspring. They're ecologically quite different from each other, and certainly phenotypically, they look different from each other. They've got different diets. Brown bears will eat a sizable amount of vegetation and fish. Polar bears prey on large mammals, including humans. They're one of the few predatory species that sees humans as prey. And despite their differences, they can hybridize, they produce fertile offspring. Brown bears from certain islands in Southeast Alaska actually have DNA that's more like polar bear DNA than other brown bear DNA. That's weird. The latest is that polar bears and brown bears are mostly distinct in their nuclear genomes. And that split happened about 600,000 years ago. But polar bear DNA is cladistically nested within that of brown bears. So if you do a cladistic analysis of mitochondrial DNA, here's the polar bear. If you look over on the right, that's the polar bear sequences. And the polar bear sequences are nested within the brown bear sequences clade, meaning the last common mother of all living polar bears was a brown bear, which is weird. And we've actually had to modify this scenario because it turned out that there's more polar bear admixture in the brown bear nuclear genomes than we thought. And it's especially found on the X chromosome. What's happened is that brown bears and polar bears split, but brown bears continued to mate with polar bears. And in particular, brown bear males have mated in the past with polar bear females, up to the point that in some brown bear populations, up to 9% of the genome originated in polar bears. And if we go back here, it turns out that it's not that polar bears are descended from a brown bear mother. Brown bears are actually descended from a polar bear mother. And that's why polar bear mitochondrial DNA nests within brown bear mitochondrial DNA. Brown bear mitochondrial DNA is actually polar bear mitochondrial DNA. It just happens to be in the bodies of brown bears. Yes, this is weird, but it's a great example of introgression where genetic material from one species can trickle into the genome of another species. We don't have any evidence for hybridization going away the other way. Male polar bears don't mate with female brown bears, but male brown bears have been seen mating with female polar bears. And this is where things start getting weird. What do you do when populations can exchange genes, but they don't exchange genes asymmetrically? This introduces the principle that I talked about a few minutes ago. Many years ago, as a young grad student, Berkeley, California has a reputation for being insanely liberal. I mean, these days, anybody that you don't agree with politically tends to get called a socialist or a communist. Berkeley actually did have a communist bookstore. And I don't mean communist in the sense of, to the political right or to the political left of Attila the Hun. I mean communist in the sense of they were actively working for worldwide revolution by the working class and an abolition of private property and all of that. They were genuine communists out there. But because it's considered so crazy liberal, the University of California tends to attract a certain population of crazy conservatives that tend to, come onto campus and scream at people. And we had this guy named Pastor Glenn, who was an evangelist. who would come onto campus and preach. You know, we occasionally will have preachers show up maybe once a year at UCA, or at least they did in the before time, before the pandemic. Maybe once a year, we'd have some group of people standing on the steps of the chapel and screaming that, you know, because we weren't saved, all of the male students were gay and all of the female students were whores, which sounds like a really bad way to go about things, because if the male students are gay, why would the female student? Never mind, not important. We got that every day. And I bring this up because Pastor Glenn and I would sometimes get into these kind of, you know, back alley knife fight debates. And at one point, he yelled at me that evolution could not possibly be true, because if it would be, there would be no way to classify living things, and everything would blend into each other, and you couldn't tell the difference between cats and dogs and things like that. We'd have no stable way of classification. And I said, that's the first thing you've said right. Because we do live in a world that's evolving, in a biosphere that's evolving. And if speciation really is happening, there ought to be lots of cases where we have two populations that are maybe sort of separate species, but maybe not. Or almost separate species, but I'm not so sure. Or mostly the same species, but you can see they're getting a little different. There ought to be a lot of ambiguous cases. And in fact, there are a lot of ambiguous cases. The biological species concept is not guaranteed to give you perfectly clear and unambiguous results. There are lots of cases where two populations are sort of different, but not entirely different. And this is exactly what we would expect in a world where evolution really is ongoing. And that's why I call this the principle of it is that sometimes when you try to answer the question, do we have one species or two? The only honest answer is, there are cases where you can't really give either a yes or a no answer, because there are so many shades of gray in between the yes and the no. Sometimes your answer is, and that's not wrong. That's the most right answer you can give. And it's the kind of answer that you would expect to see on a world where the biosphere is always changing. A few more cases of before we have to end. I see I've got about 5 minutes. You can't readily apply the biological species concept to asexual organisms. Many amoebas have never been observed reproducing sexually. If they're doing it, they're extremely discreet about it, but we've never caught them. A microbiologist named Sorin Soneo once observed that if you follow the biological species concept, there's only one species of bacterium, because bacteria can pick up DNA from almost any source. And when bacteria have sex, that's what they do. You might remember the transforming principle experiments of the 1920s that showed that living bacteria of one strain could pick up DNA from dead bacteria. That kind of thing happens all the time. Bacteria can transfer genes all over the damn map. So if we go with the BSC, there's only one bacterial species in the world. And then we sometimes get answers that seem incongruous. Here we've got eastern blue jays on the left. And in California, I used to see these guys on the Berkeley campus. They're quite attractive. The males have those beautiful black heads with blue feathers. That's Steller's jay. They look different. They live in different areas. But there is a small part of Colorado that is what we call a hybrid zone. In that zone, they overlap, they interbreed, and they produce fertile hybrids. Meaning, I suppose, that genes from one could very slowly introgress into the other. So do we have one species or have we got two? On the other hand, we have cryptic species, and I mentioned this one. Western and eastern meadowlarks, physically you can't tell them apart, but they don't interbreed. They're behaviorally isolated because the males sing very different songs. So this is another case where the biological species concept does not line up with what you would see in a bird watcher's manual. Here's another case. This is Kirk's dictic. Insert giggling here. It's a small African antelope, and there's three populations that have radically different karyotypes. They differ because of chromosome fusions and inversions and translocations and all that. Crossing individuals with different karyotypes produces sterile hybrids, even though you can't tell the difference by looking. I'm going to go ahead and stop there, and we'll talk later about California wildflowers called Farewell to Spring. We will pick that up next time. So I'll go ahead. This is a pretty good place to pause. So I'll go ahead and stop the recording.